You want to start it? Okay. So let's start one more time. Welcome everybody to the Pages Early Career Network North America Show and Tell series. My name is Sandra Brugger. I'm one of the North America representatives of the Early Career Network and a postdoc at the Desert Research Institute in Reno, Nevada. Thank you all for attending today's Show and Tell event. So today we are joined by Dr. Robin Dawson to learn more about her research on speleothemes in Madagascar, and I'm super excited about it. Robin is a postdoctoral research associate at the University of Massachusetts in Amherst. She did first an undergrad at Macalester College in geology, followed by a master's degree at University of Wyoming, and then she got a PhD from Yale University in geology and geophysics in 2019. Robin is interested in paleoclimate, paleoecology, carbonate chemistry, organic chemistry, and stable isotope geochemistry. Robin particularly loves doing fieldwork, experiencing new places, and meeting new people. Though she also enjoys the zen state, as she says, when she gets in while she's doing repetitive lab work. And she's also the mom of a rowdy toddler. Robin is going to present some of her research on Holocene environmental records from speleothemes in Madagascar. She will talk a bit about her work attempting to use organic geochemistry techniques on speleothemes to inform about vegetation shifts above caves, as well as the more traditional work at UMass using stable isotopes of cave carbonate to reconstruct changes in past precipitation. As always in our show and tell series, you can write your questions in the chat box, or you can also raise your hand and then unmute yourself and directly ask your questions. And with that, Robin, I would like to give you the floor, and I look forward to learn more about your research.
Great, thank you. And thank you for the whole Pages Early Career Network for inviting me. I'm really excited to share what I've been doing for the last couple of years. Let me just pull up my PowerPoint. Okay. There we go. Okay. So I'm assuming everyone can see this. Just quick thumbs up.
Yes, we can see it.
Okay, great. Thanks, Maddie. So yeah, thank you, everybody. My name is Robin Dawson. I am a postdoc at UMass Amherst, and for the last 2 years or so, I've been reconstructing environmental change from cave deposits in Madagascar. And luckily, right before COVID, October 2019, we just squeezed in a field season right in time. So I actually got to go to the cave that I'll be talking about in this presentation and sample the speleothem that I'm working on right now, which is a really cool experience. And so for those of you who might know a little bit about Madagascar, one of the really exciting things about it is it's been fully isolated from the mainland of Africa since the Cretaceous. So it's the 4th largest island in the world. And because of that, it has this like really interesting endemic flora and fauna that have just been like evolving and doing their own thing for 80 million years or so. And so because of that, a lot of the economy of modern Madagascar, formerly the Republic of Malagasy or Malagasy Republic, is this like ecotourism and sometimes like high-end ecotourism, safari type vacations. So that's a lot of their economy. So there's all these cool animals there, but unfortunately, there's also a lot of wealth disparity on the island. A lot of people are still subsistence farmers, heavily focused on rice. And recently there's a lot of news. I don't know if people have been hearing about this, but global warming is really affecting these poor tropical countries. And Madagascar is one of those, experiencing a lot of droughts and then heavy, devastating tropical cyclones. So, not a sort of reliable precipitation to do farming. So this is sort of one of the motivations we have for our research into paleoclimate in this area, to learn about past changes in precipitations for vulnerable areas for future global warming. But just sort of more generally, I just have a little video. if you look at sort of the global record of paleoclimate proxy records, they're super biased to the northern hemisphere, especially the tropical zones. Ocean cores have really good coverage, of course, but if you look at terrestrial paleoclimate, the southern hemisphere, there's a lot less data there. So we're interested in things like, how did these big global events like northern hemisphere glaciation, how did that affect the tropics? How did that affect the southern hemisphere in particular? And then in addition to that, because Madagascar is this unique place for life, for plants and animals, we're sort of interested in reconstructing climate context for sort of changes to animal life, plant life, sort of microcontinent of Madagascar. And so just here's a summary of Madagascar. It is really the main features is sort of eastern highlands here, the sort of north-south trending mountain range. And that sort of divides the island into these two zones. And so you have a lot of orographic rainfall happening here on the east. And then basically the rest of the island is just in this rain shadow, and much drier over here on the west. And then sort of this north-south pattern of more rainfall, a lot drier down here in the southwest, that is a result of the other major controlling climatological feature of this region. And that is the migration of the Intertropical Convergence, or monsoon, over Madagascar. So Madagascar, other than that orographic rainfall, gets most of their rainfall in the austral summer, as you can see over here on the left, and then it's really, really dry the rest of the year. And so this is sort of how the system works today. We're curious in sort of big questions like, how did the monsoon change in the past? Sort of that north-south gradient of wet, dry, like how did that change in the past? And this will really, I think, help us give some, guidelines and idea for how to advise people now who depend on agriculture today. And so the site that we get our paleoclimate archival material from is this cave in the northwest here. The biggest city that we can get to this area from is Majunga. It's like the biggest city on the west coast. But this cave is called Enjoy Bay, which means big cave in Malagasy. Enjoy Cave Bay, big. And this has been a site that has been published on before. For example, Wang et al. 2019. This map comes from. But it's an absolutely beautiful cave, and I just felt very privileged to be able to go there. We had a wonderful Malagasy guide, Dawson. He's down here on the left. We actually we have to give a sort of offering before entering the cave out of respect, and so we're giving honey as our offering. But yeah, just beautiful large caverns, which some of our samples come from. I'll show pictures of that later. But there's actually this underwater stream that we got to float through, which was amazing, and hollowed out canoes. Absolutely beautiful cave. And so here's an image of the sample. I'll actually be talking about it in more detail. You can see it's kind of, there's a hammer for scale. It's about 2 meters long, probably a little bit more if we had been able to get that based, but didn't really. But you can see there's sort of like a thinner, more narrow diameter tip up here at the top, roughly 30 centimeters or so, and then it gets much wider. This was sort of a beast to sample. I wasn't sure we were going to be able to do it. My PI had some crazy ideas to break up like a pulley system with rope at one point, but we actually managed to just sample it with a hammer and chisel in the end, which is sort of the main tools of a speleologist, I guess. That and a helmet, of course, for safety. But it was so heavy, we actually had to take a fallen tree and carry it out, tied to it, this tree with rope. So this was no small feat. And then what, of course, I'm not showing here is just sort of all the paperwork and, bureaucracy and sort of relationships you have to build with the people there, the scientists there, and the communities there, because it's actually a big deal to come in here and sort of interfere, and then you want to give back to the community in some way, and so that's really important to us in our work too. So just to take a step back for a second, for folks who maybe aren't familiar with caves and speleothems and how they work and how we can get climate data from them, just sort of a quick cartoon. Basically, we're measuring these cave deposits that form underground, and we're measuring stable carbon and stable oxygen isotopes from them, for the most part. And they record information about precipitation. So water will rain down, percolate through the soil and the karst system, and it'll pick up atmospheric CO2 along the way, and soil CO2, so respired from roots and decomposing organic matter. It'll mix with water, forming bicarbonate, carbonic acid, and then dissolve, flow through the system. And then once it hits the cave environment, the partial pressure of CO2 is actually lower within the cave than what was in the soil, the atmosphere. So you get degassing and then actually deposition of the dissolved carbonate, usually as calcite layers. So that's what this little inlet instead is here. And so for the most part, the water is just reflecting precipitation. This is a function of the source, so where that air mass is coming from, a little bit of temperature. But in the tropics in particular, this is really reporting the amount of precipitation. And so if you have more enriched values of delta 18O, you have less precipitation. If you have more depleted values, you have more precipitation. And I'll show a graph to show how this works next. But the other sort of climate or environmental signal you can get from the calcite stable isotopes is the carbon, which can reflect overlying biomass. And so, whether it's C3 trees and shrubs or C4 grasses, you'd have more enriched delta 13C if you have more C4 plants. So that's just sort of the quick summary. Obviously, it's much more complicated than this, but again, the sort of main thing we're getting when people are looking at speleothem records, delta 18O, that's what they're interested in, and it's this showing this amount effect. So you can see this is just New Delhi, which is also a monsoon region. So this is why we think the amount effect is the overriding control in Madagascar. You can see that in New Delhi, when you have more rainfall, you have more depleted delta 18O. And so we're using this relationship to interpret the record. Now, going back to how we actually do this, this is actually part of that same sample I showed. It's the thicker part after the top 30 centimeters. On the left, I'm trimming it actually with our tile saw, which was actually too small to cut the whole thing in half. So when we bring these things back from the field, we cut them in half so we can see those growth layers that form through time. And we actually had to cut this sample and a couple others because they were so big in half. We had to go through to a local like stone quarry for building materials. They mostly cut local schist and slate for houses and stuff. And you can see this giant saw we had to use to cut these things. So it's kind of fun. And another big part of this, and then sort of the first step do is to get an age model on these speleothems. And so we work with colleagues at MIT to do this. PI David McGee, and I've been working with a PhD student, Ben Tiger, who's been doing the uranium thorium age model for us. And this is just a really rough age model. For now, we're waiting on 10 more samples, but it's helping me as I gather data that I will show, which is like data generated last week or so, just to give me an idea of sort of interpreting our initial results. So this top little 30 centimeters that's much more narrow, we're getting, planning my sampling by depth based on this. So I'm assuming every millimeter or so is about 9 to 10 years. And then when the sample gets thicker, when this sort of age model changes, I'm sampling at sort of lower resolution because now it looks like based on the age model, we're getting every millimeter 2 to 3 years. So I'll show you in a bit, like this sounds really simple, but it's actually sampling these things. One, it's very time consuming, and that Zen state is very important, but you can get a sense here of like how the growth axis shifts a little bit. But that's sort of, I think, one of the things maybe we don't check that as much as people who study these records. I just wanted to show some of the cool toys we have here that aren't in this physical lab that I'll walk around in a bit. This is down the hall. It's the XRF core scanner, the iTrax. It's actually designed for scanning lake cores, but what we've been able to do is actually put our cut in half speleothem into this cradle here you see with a yellow measuring tape. It's about 2 meters long. And we can program this XRF overnight to just scan, like it would a core, scan our speleothem every 0.5 millimeter, and we get all this elemental data overnight. It's amazing. So things that will take me months doing oxygen, carbon isotopes on the gas bench, I get this overnight. It's amazing. And we're interested in some of these trace metals because they can sort of help corroborate our paleohydrology data from the stable isotopes. And one of those is strontium. So strontium calcium ratios are often used to compare to the oxygen isotopes to look at wetter and drier periods. The idea is that during drier periods, you have a longer residence time of the water in the karst system. And so the calcite is actually able to take in more strontium, which it doesn't usually like to do just based on the ionic size and the crystal lattice of the calcite. But if it's hanging out there for longer periods of time, it picks up more strontium. And so during drier periods, you have these higher strontium calcite ratios. I'll show this lab in more detail in a bit, but basically we drilled the sample, powder it, we put it in a vial, we put acid on it, we flush it with helium, we load it up in a gas bench. I can walk through that in more detail in a little bit, since that's where I'm standing right now, in this other half of this lab. But the data that I've been generating in the last week or so are this top, basically tens of centimeters of that little thin part of this speleothem that I've been showing you. And so it's been very exciting. My first gas bench data that I've generated while here at UMass, because I've been working on this other project for so long. But there's some interesting things happening here that's maybe sort of an abrupt drying period and then back to wetter conditions. Again, I'm just showing depth here on the x-axis because our age model is still getting worked out. But as I generate more and more data, I'm really excited to sort of see what sort of centennial, millennial patterns we're picking out on this sample, which goes back 10,000 years or so. And ultimately, once we run the whole sample, I'm curious in comparing it to other records. So my hiatuses are off, but this published record from Wang et al. 2019 has some hiatuses, so we're hoping to maybe fill in some information there. And then we're also really curious to compare this basically like full Holocene record to the southwest of Madagascar. This is a record of the last 4,000 years that was recently published by our Malagasy PhD student, Peter Santana, in Malagasy Nature. So he had some cool interpretations about what was happening with the megafauna extinctions down south. Sort of other larger scale questions we want to ask with this record is comparing Madagascar, which is roughly 15 degrees south latitude, the northwest, to the tropics on the other side of the equator. So Oman is roughly 15 degrees north. So we're curious, like, what is happening with the main tropical rainbow? There's some evidence that it shifts south during periods of time, particularly during when there's large continental ice sheet. There's evidence from other records that it sort of expands, and actually the northern and southern hemisphere are in phase. They're actually doing the same thing, like parts of this record from Stroxton in 2017. But then it's out of phase during other times. There's this big drought. recorded in a lot of sites in the Northern Hemisphere at 8.2 thousand years. There's a China and Oman record from speleothems, and it's dry there, but it looks like it's wet here in Madagascar and Brazil in the Southern Hemisphere. So those are some of the sort of the bigger picture things we're interested in our sort of traditional, speleothem bread and butter proxies, which is calcite delta 13C. However, prior to this recent spring, a lot of my work has been looking at these less traditional proxies that aren't often used in speleothems, and we've been trying to develop them so that we can add some information to this question about the Holocene loss of large mammals in Madagascar. So this is just a little schematic showing, Africa still has these large megafauna today living, but other continents really lose their megafauna. Here, this is the green curve, right when humans evolve at the black arrow. In Madagascar, however, the megafauna loss is much younger, or is much later, more recent, and there's actually this larger gap between when the humans arrive and when the megafauna go extinct. And so we're kind of curious what's going on there. And so for a while, there's been research into, were there climate drivers, were there human drivers for the extinction, and sort of what was happening. Some of the cool stuff that was living back then are these giant elephant birds, these large, so the fossil lived there today, but there were these large giant fossa back then. There were the crocodiles. There were all sorts of different types of lemurs, more than the types that live today. There are even pygmy hippos. They're up here in the corner, tortoises, and they all go extinct. And so the sort of existing hypothesis right now is that this is the subsistence shift hypothesis, and this is work done by our anthropologist collaborator, Lori Godfrey, here at UMass. And she compiled a bunch of archaeological evidence from fossil bones, which had cut marks on them, and radiocarbon dated them. And she saw, evidence from archaeological sites that the human population was sort of steady and then rapidly increasing. And that sort of coincided with this time where a lot of the fossil that they were dating were these extinct taxa, and then a mix of extinct and extant taxa. And then all the younger fossils they're finding are all the extant taxa. So this sort of lines up also with a delta 13C record from Anchovy Bay, where there's a sudden enrichment in the delta 13C, suggesting much more seafloor plants on the landscape. There's not a lot happening with climate at this time. It's pretty stable. And so what the authors think is happening, what we think is happening, is in Madagascar, unlike these other places like Australia or North America, is that humans were sort of in Madagascar, but they were sort of ephemeral. They were like using it as a jumping off point or a temporary settlement, and they were predominantly foraging and hunting. But the real change happened here, around 900 CE, and they became a much more stable population in the region, and they switched to farming. And in particular, this practice of tabi, which is kind of like slash and burn agriculture, where they burn the landscape to promote the growth of certain grasses. And so that's what we think is happening here. Here's just a skull. This is a really cool Megaladapus, is this lemur that was sort of like koala size. Some of these like lemur type things are so cool. Anyway, so we're trying to basically catch this C3 C4 transition in the delta 13C of carbonate in Anchovy Bay, but we're trying to see if we can catch it with a record that's more tied directly to organic matter. So delta 13C of organic matter, or delta 13C of biomarkers, like molecules that directly reflect plants, like leaf waxes. And so, as you can imagine, there's not a lot of organic matter in a carbonate. And so I need a lot more carbonate to be able to get any sort of signal. And so you can see this picture, and I'll show you in a little bit. I need around 10 grams to do that, and I use this core drill bit to do that. So sort of the struggle I've had in doing this project, and I think it's important to talk about this type of thing. You know, a lot of times in science, it's like the flashy end result is shown, and we don't really talk about the struggle of, like, in particular, developing a new method. And so that's what I've experienced. And part of it was that a lot of the published papers showed that this was possible to measure delta 13C and specific biomarkers of plants or other things. But when I tried it, I wasn't getting results. And I thought about it for a little bit, and I was like, well, these published methods are relying on cleaning the outer surface, because we have like waxes on our fingers, so we can't like touch these things. When you saw them in half, there's maybe stuff in the water from the saw that lubricates the saw. So how can we clean the outer surface, especially when the concentration of organic molecules is going to be really low? Contamination is a real problem. But these published methods involve sonicating a chunk of speleothem. And I'll show you, my speleothem is like pretty porous. So I realized there's really no way the solvent's going to differentiate sample from outer contaminants. So I've sort of been developing this new method where instead of doing that, we're polishing off the outer surface with a diamond studded polishing plate. And then we're basically following the existing methods, which is dissolving the chunk of speleothem in acid so that we can recover more organic matter. It's produces a better yield than just sonicking or extracting the sediment, like sort of powdered carbonate, which is what you would typically do with like core sediment. And so to get this higher yield, we then have to liquid extract the acid, which now has fully dissolved the carbonate. Any organic molecules within the carbonate layers is now in the acid. We have to extract the acid from a solvent, and now our sample is in the solvent. And then we can just use the existing methods that have long been used by people who study ocean cores and lake cores, column work, GC analysis to separate out these molecules and measure them for pseudoisotopes. So I just wanted to highlight this a bit because I feel like this is something we don't talk about, method development and how hard it can be. But I've presented some of our initial results on this at AGU, and we have found that we just, in terms of nanograms of N-alkanes, which are a class of these plant waxes, we just get way more when we polish the outer surface than when we sonicate clean the outer surface. So my hunch that we were losing sample is probably right. And also, this is just to show again that the pale tan here is the solvent we use to clean like my drill bit and tools that I use. So just showing you how important it is to clean stuff before you use it and how careful we need to be with contamination again. But we're getting some promising results. We are seeing, you can see here in the orange, this is the sample that we polished clean. There's a bit of an odd even predominance, which is what you would expect in the actual leaf wax. And then looking at sort of diagnostic ratios of leaf wax carbon chain lengths, C31, C29, this sample that I've been testing on, so all of these samples here, but looking at the orange because that's the polished samples, they're giving us ratios that reflect more woody angiosperms than grasses. And this is actually what we'd expect because the practice sample I've been working on it doesn't have a great age model, which is why my PI was okay with me playing with it, but it's almost certainly older than 2000 years, so before the C3, C4 transition. So it should reflect more C3 plants. So this makes sense. And then also I've just been looking recently at characterizing some of the plants I collected while there that grew near the cave and near the area. And yeah, if you look, I have mostly C3 plants here, a grass that's a C4 and a cactus, which they actually grow these cactus. It's kind of interesting. They grow these cactus for the zebu or the cows, because in really dry periods when the grass isn't growing, the cows like to eat the prickly pear. So it's kind of cool, but sort of human brought for taking over. But you can see today the chain lengths of sort of the modern things on the landscape are closer to more C31 than C29. hoping maybe we can see this shift in chain length that coincide with that enrichment in Delta 13C. Have been able to get a couple of, you really need a lot of materials, so we haven't been able to get it on every sample, but have been able to get compound specific Delta 13C on some beef waxes, fatty acids, methyl esters, and N-alkanes. And you can see that down here. I've also measured the total organic matter extracted through that liquid extraction, and they're giving us Delta 13C values that look like C3 plants. So all what we sort of expect for older than 2,000 years ago, before humans got there. And that's basically it. I have a couple more photos of organic method stuff if people care, but I just like to say thanks. And I guess I'll walk around the lab for a little bit more detail and then come back for questions, if that makes the most sense.
Yeah, thank you so much, Robin. I think it would be very interesting for us if you can walk around with us a little bit. And
yeah, let me switch to my phone so I can be more portable. Okay, great. So I'm just going to walk around the lab a little bit and show you. Well, so here's my sample. Right now it's on our micro mill. As I said, the data I showed you was like fresh off the press. So we have, you know, a calibrated base that we can basically sample every millimeter. I think I use a like 0.3 micron drill bit on this thing, but it's been a little tricky, and I'll show you some sections later. It's been pretty straight measuring the sample down the middle of the growth axis. These larger troughs are where we take larger powder for our uranium thorium age models. But you can see sort of around here, I don't know, there's a weird, you know, the way we sliced it in half, or there's some recrystallization or something. I had to sort of get a little creative. You can sort of see my sampling path gets, yeah, a bit different. And then I'm back to down the main growth axis. But so this is the sort of Zen state I was talking about. It's pretty easy as you just crank this dial. This can tell you, you know, every millimeter and then drill. But it gets tricky when you get to these regions where something funny happens with the growth axis. So this is that top narrow bit. And then over here, I have laid out the rest of the sample, which I'll be working on in the next several months, weeks. It's quite a bit more. You can see we've had to trim it on the back, basically on every side. I can show you the other half. It's still our archival half, which we like to keep for things like the organic processes later down the road. You know, we do the traditional age model and Delta 13C, Delta 18 on the carbonate on this main half. And then down the road, if, you know, new proxies get developed and stuff, we have the archival half to do sort of more creative stuff with. But so this is where it gets thicker. This is actually the base of that narrow part. It's going to be interesting too here. You can see drilling this, because I've been talking about this with my PI. What we're thinking is happening is that the main growth goes to about here, and you can see this sort of like brown line that overlaps both of these parts. And we're thinking we've cut, you know, because this isn't, I mean, you sort of schematically model it like a perfect cylinder, but it's not really like that, right? So what we're thinking is we've sort of sliced it in a part where we're seeing more of maybe this than this. So we're thinking then we drill down here, and then with this large gray or brownish band, pick up with it here. So it's kind of not a straightforward depth growth axis, but that's just sort of the fun things you find out when you cut your sample in half. But otherwise, there's sort of the typical changing of the growth axis as the speleothem grows and sort of breaks itself for balance, and that's sort of more expected. But yeah, so that's my sample. That's my baby, other than my actual baby. And now, well, so this is the other half of it. It's quite hefty, as you can see. We had to cut off all of these edges. This is just half of it. The other half is in this tub. Yeah, this is sort of the sample processing side of the lab with our mill. And we actually got this. This isn't really something I've been using, but this just got delivered. This is actually a computer programmable micro mill. And I'm not exactly sure how the other group is going to be using this, but right now they're testing it to try to program the drill to run this whole curve on the growth layer of this shell. So we'll see what we can develop with this new toy, but so far I don't think we'll be using it for a lot of speleothem work. Yeah, and then this is the louder. You thought the other room was loud. This is our even louder side of the lab where all our mass specs live. And so we actually have this delta that's hooked up to the gas bench, which is where so these are actually those last probably 70 samples I showed you in that figure. Still waiting here, I gotta clean my vials. But you know, our auto sampler can run overnight. It's really nice. We put some standards in, but you can fill this thing up and get like 80 samples run. So after we powder the material, we sway them out in these vials. I feel like probably a lot of people have worked with gas benches before, so I don't need to go into too much detail with this, but you know, seal them with caps. We have you know, really pure more than 99% phosphoric acid in there that we inject with a syringe. But prior to that, we flush all of our samples with these syringes, actually sort of designed in-house by our machine shop guy, who's great for this exit hole. He designed this to flush the helium out. And then we can load them up there in the tray and inject the acid, and this thing runs overnight, which is great. And then the other side of this lab is actually our GC IRMS. We actually have two of them. So I also work with Isla Caponeta on this project with the organic molecules, and she's got this, I think, that can run carbon or hydrogen if we want. Basically, we can run carbon or hydrogen on organic molecules at the same time if we want. So we have another delta over here, brand new, that runs this. And so the carbon isotope values of the fatty acids and the N-alkanes I showed you was all run on this instrument. Brand new, really nice. We just got it like the beginning of COVID. It's sort of bad timing, but it all worked out in the end. So actually someone has something pulled up here. I think someone was picking hydrogen isotope peaks on some N-alkanes, but yeah, you can sort of see her chain length peaks over here. But yeah, that's basically our lab. This is the other half of the sample I've been playing with. So it's about the size of the sample that generated that. Let me turn back around. So, oops. It's about the size and was found near the sample that was published, where we see that large enrichment in Delta 13C. So that's why I've been playing around with it for the organic project. But yeah, as you can see, I mean, I've been trying this method with the polishing plate. It's just this, you can buy this, I think we bought it off Amazon, and these diamond tip drill bits, which we have even larger ones that I've tried even a larger core drill bit for playing around with the sample, but obviously very low resolution. But when you have such an abrupt large signal, I think this type of method could actually work. And I'm playing around with a marble too for a standard, so drilling giant holes in that. You'd think I was doing paleomag or something, but I'm not. So yeah, that's that's basically it. I don't know if we want to do questions now or, yeah, I'm open, whatever. I just want to say thanks though, again, this was really fun. And it's been so nice to have this to look forward to. I think a lot of us with COVID were like starved for networking and social interaction. So I really appreciate this chance to share this all with you guys.
Well, thank you very much, Robin, for giving this interesting presentation. It's awesome. Yeah. Well, guys, please all feel free to ask questions. And if not, I want to start with a very easy one. I mean, like, you're essentially like handling rocks. How heavy is that as a workout?
Oh my God. Yeah. So I showed you that picture, right? Let me maybe move back to my or stop video here at least. Well, I'm like, you know, a feminist and I want to be hardcore and whatever in the field, but I will admit you'll notice in that figure, I don't need to show it again, I guess, but it was definitely just the guys carrying the thing. So it was me and a woman who works for the Department of Mines in Madagascar, and she was with us just to make sure there was a representative from the government, you know, making sure we were doing all the right protocol. And it was actually great having there because she was so helpful and knowledgeable, and like she's a geologist herself, and so just having her insight was really cool. But yeah, I will admit, she and I were just like, you guys got this. We're gonna hang out in the cave and explore a little bit. We saw a turtle in there, had wandered in through some of the crevices. But yeah, we had to lug it into an SUV. You know, all the typical field stuff happens. The SUV, you know, our cars like blew a fuse and our engine broke down, you know, on the side of a cliff, and we're having to like move the speed ascent. We had 2 SUVs and move all the samples from one to the other while someone waited for help. It was, you know, a lot.
Well, if no one else has a question, the other thing I was wondering, like you said, you polish the surface of your samples, and then do you have to like re-polish it every time, like when you drill to get the samples out, or when you like before you run XRF, or can you like polish it one time and then you're kind of like good?
Yeah, I forgot to show you our polisher. Yeah, we polish it one time and we're kind of good. So we use this wet polisher. I mean, it's basically like, we look like we're installing like bathroom tile with our tile saw and our polisher, but it has, you know, different grit plates that we can attach to get. It is nice to get a good polish. And yeah, usually you do one and done and you're good. You're sort of touching on something that we do with the XRF. We want to scan it before we drill it, because the different depths of the pits can actually affect the XRF. So that's why it's nice because it's fast. We can do that before we start drilling for stable isotopes, and we can sort of get a bunch of data really fast that gives us a sense of the record we might produce. But yeah, we don't have to repolish it typically.
Cool. I see Shivanji has a question. Do you want to unmute yourself?
Hey, hi. I'm not very good with this, so this might be a really silly question, perhaps. It's
silly question.
I saw that your the 13C pattern and the 18O pattern is very, very similar. But
yeah.
Are you? So I read up a bit of speleothem literature a few years ago. So I don't know if this is, you know, how do you make sure that they are reflecting, you know, the vegetation or the precipitation and not something that is to do with the cave environment?
Yeah, I mean, you've hit on like half of, I feel like, speleothem paper publications are actually not on paleoclimate and on like karst processes.
Right.
So yeah, I mean, mostly the delta 18O really doesn't change for the most part when it percolates through the karst system, and then, you know, you have the dissolution and precipitation. The carbon is a little trickier. And it's actually pretty unique that we interpret in this region a vegetation shift, because in a lot of places, like the observation you noticed, more often than not, the carbon isotopes are doing almost exactly the same thing as the oxygen, and there's nothing really different to interpret. And it's most likely reflecting, the same thing, wet or dry, but just filtered through the vegetation on the landscape. The plants are plants that like wetter conditions or drier conditions, or the plants are water stressed, things like that. So it's really the carbon is reporting sort of a similar, although through multiple filters, information as the oxygen is. When they don't do the same thing, which is what was happening in Anchovy Bay, like oxygen isotopes were basically kind of flat, and then carbon was just like, boom, 10 per mil enrichment. That's sort of when we were like, maybe this is really something strong, like something more direct is happening here. And with all the other evidence from the area of like land use changes, we really think it's vegetation. But it's not often that Delta 13C is really interpreted a lot or in that way, I feel like, in paleoclimate records.
Right.
Because there's all these complicated processes about like rate of degassing and disequilibrium effects and stuff like that.
Okay, thanks.
Robin, I'll ask a question.
Yeah.
The cave, is it also a tourist destination? Do people go in this cave
Oh yeah.
and interfere?
Yeah, they do. And unfortunately, I mean, I didn't want to show it, but I do have a picture like of cave graffiti. So it is Anchovy Bay. There's an awesome, and if you're ever there, the Grotto de Anchovy Bay campsite. I forget exactly if it has another name than that, but there is a local family that runs like camping huts. They're actually really nice. They have flushing toilets in these like grass huts, and they cook dinner for you, and there's like bonfires every night, and a guy playing guitar. And yeah, it's definitely tour. It was like the cushiest field work I've ever done. Not camping. We were like sleeping in a bed with flushing toilets. But it's definitely, there's a lot of French tourists because, Madagascar used to be like a French territory. And so there's a lot of French tourists. There's a lot of like birders and people who are into nature. It's not developed tourism, but it's like this eco-tourism, which is really strong there. So you definitely go. You just need a guide. And I think it's important too, because that is a big part of their economy. So you can't really just like go there and drive yourself. Like, you can't do that.
Do we have other questions? You can also type them in the chat if you want to.
I will say I'm new to speleothems, so I had worked mostly in the marine realm and in the Cretaceous prior to this. So no questions are silly, because I've asked a lot of them in the last couple of years.
Yeah, actually, I have one more.
Yeah.
So how did it feel going from the Cretaceous, which probably seems like real deep time or real work, to the last millennium? I think that's what you're targeting right now.
Yeah. And like, you look at the Vostok core, you look at like this record, and you're like, the Holocene, what's happening in the Holocene? Like, there's maybe that 8.2 event, and then, for someone who was looking at deep time, I was like, what would be interesting about this? And I've been really surprised, actually. And actually how much similarity there is, because when I was studying the Cretaceous, I was interested in like temperature gradients and cross hemisphere, movement of heat and moisture and precipitation. And so thinking about it now on these shorter time scales and sort of much more like dynamic processes, I think really complements it. And I've always, been interested in, like, in the Cretaceous, it was like ammonites and dinosaurs. And so my sort of love of paleoecology, I have really been able to find exciting all the anthropological work and the tie-ins with the archaeologists that we do has been really cool. And then just like being able to do something that feels a little more not applied, but like imminently relevant with anthropogenic global warming. I mean, like, people are starving there, and not that we're gonna save the day. I mean, it's gonna be like engineers and farmers who develop new techniques for farming or whatever. But I think just getting the word out and being, an advocate and like a scientist who can speak to the public and like stress how important this stuff is, that feels good. So moving into the Holocene has been different, but I find it really rewarding. Let me see, there's maybe a comment in the chat. Yeah, I totally agree. I think this is the other thing I felt really important. Like, I felt really privileged to do this work. Was just like how biased scientific work is. It's like a lot of it is done in countries that have the resources, and there's all this information we're missing. So yeah, thank you for that comment about, I would love to extend this work to West Africa. Be really interesting too, because with our questions about things like this drought at 8.2 thousand, which we think was like triggered by freshening of the North Atlantic, it would be really curious to see like these sort of distant and then maybe an intermediate point with West Africa. Like, is something happening in North Atlantic impacting the Western Indian Ocean and Madagascar, and like what other regions in the tropics, like how does it affect it? I think that would be really cool. Or does it? Maybe it doesn't.
Well, good. I think if no one else has anything to add, I would like to thank Robin again for presenting today. It was amazing.
Yeah.
Please
No,
give
thank you.
Robin a last virtual applause. And then I thank you all for joining us today, and also to learn about Robin's Philatem work. And most importantly, if you haven't signed up yet for the Pages Early Career Network, please register to receive information about many different networking meetings with other Pages Early Career researchers. I think Maddie posted the link in the chat. So stay tuned for our next Pages North America show and tell events that will be announced soon. And with that, I would like to close, and thank you again very much for taking the time to joining us today. Enjoy the rest of your day. And thank you so much. Bye.